Hello, everyone, and welcome to our monthly seminar series from Medric, the Medical Imaging and Data Resource Center. Today, we are happy to have Dr. Sharon Katz. She's a thoracic radiologist and director of research for the Division of Thoracic Radiology at the University of Pennsylvania. Her research is focused on and devoted to thoracic oncology imaging for detection, staging, response evaluation, and treatment guidance for thoracic cancer. She is happy to present to you today on leveraging registry data to conduct virtual clinical trials, part of the CRP Clinical Research Project number 7 of Medric. She has, besides being a physician, she also has a master's of translational research. I found that very interesting, and I look forward to her presentation today. Dr. Katz?
Thank you very much, Mary Ellen. I'm going to share my screen. Are you able to see?
Yes. Do you want to go into slide? Okay, good. Great.
All right. Well, thank you for having me today. It's a pleasure to be with you today to talk to you about our project, leveraging registry data in virtual clinical trials. And this is the COVID-19 data that's being collected as part of the Medric effort. So just as a quick review, many of you probably already are familiar with Medric, but it is a imaging data resource that is a collaborative effort of a number of institutions as well as, and it's being hosted by the University of Chicago on the Gen 3 platform and being supported by the NIBIB with leadership from the University of Chicago, from the ACR, the American College of Radiology, the RSNA, which is the Radiologic Society of North America, and the AAPM, the American Association of Physicists in Medicine. This resource is a integration of imaging and clinical data, and the idea is to accelerate the research and development in the field of imaging. In particular, having a big data set is of particular importance to development in artificial intelligence, machine learning, and radiomics, all of which require big data sets that are curated with clinical data to correlate with the imaging in order to adequately power these efforts. So the initial project of the Medric is to collect the data, imaging and clinical data on COVID-19. And this is effort began in the setting of the pandemic, and it was the funding from the NIBIB supports 5 technology development projects and 12 research projects. There is participation from 23 institutions across the country, and with a number of scientists, physicians from in all different areas of the field coming together to really collaborate on these projects. So in addition to providing a resource for imaging and clinical data that can be publicly accessed, it also brings together these communities to accelerate research and development. And it's a very rich and fruitful collaboration. So there are 12 investigator-initiated projects that are on the initial wave of funding through the of Medric, and our project, leveraging research data to conduct virtual trials, is one of those. And I co-lead that along with Caroline Childs from Wake Forest. Just to review quickly how the data gets into Medric so you understand, there are a number of different portals. One of them is the American College of Radiology, and I'm involved in that effort, and that's collecting data, imaging and clinical data into their data platform, and then that data enters Medric through a gateway. Same is true for RSNA, that they contribute their imaging and clinical data through a gateway into the Medric, and then there are other channels, including the TCIA. This public resource is then available for access to academia, to the FDA, and to industry users. Just to say a few words about our COVID-19 registry at the ACR, which is contributing to Medric. This is the data set that we're using for our initial efforts with the virtual clinical trial effort. The ACR COVID Imaging Research Registry was established right around the beginning of the pandemic in about April of 2020, and it's a joint effort from a number of societies, including the Society of Thoracic Radiology and the ACR Data Science Institute. And the goal is to provide this a platform for clinical integration of clinical and imaging data. And we began our data collection on January 1st of 2020. So collecting data leading up to the pandemic and then continuous from then. So it's a longitudinal database beginning from the inception of the pandemic, following forward, integrating both clinical and imaging data. Our group is, this registry is directed by, I chair the committee, the steering committee for the CIR, but this is a group of approximately 30 physicians from over 20 institutions in 14 states representing 8 societies. So it's rich in perspectives and experience, and we really strove to bring different areas of the field with expertise in COVID-19 to the table to be sure that we were collecting data that was relevant to the field. And that's a dynamic process. So we're continuing to meet and kind of be sure that we're collecting the data that's necessary to keep this registry pertinent to questions that are ongoing. In terms of how we designed our registry, as I mentioned, it's both clinical and imaging data, both integrated on the ACR DART platform. And so there are CT, MRIs, and chest x-rays, as well as ultrasounds, PET-CT. The majority of the data for the COVID-19 registry is chest radiographs. There is also a large number of CTs and then a mix of additional modalities. The vast majority of these, as you would imagine, are chest, so chest x-rays and chest CTs, but we also have cardiac, which is pertinent since there is activity in this disease in the heart, and neuroimaging. And as we know, there's certainly significant neuro findings as well in this disease. We're also collecting a wide range of clinical data, so demographic data, including zip code information, although that's de-identified as it enters Medric. We are also age and sex and insurance status, medical histories, comorbidities at diagnosis, changes in diagnosis over hospital stay, and then also degree of a level of care during hospitalization. So were they admitted to the ICU, were they intubated, were they put on ECMO, and then information on long-term effects. So were they discharged home? Did they go to a rehab facility? Did they die? Also collecting information on their medications and changes in medications at onset, as well as tests of function, including lung function tests. So for the CRP7, which is a virtual clinical trial project, we put together a team to provide both the kind of clinical perspectives as well as the radiological aspects. And we have some participation from the data scientists at the ACR and a statistician, Keith Mueller, who together with his partner, Tian Hao, is providing statistical analysis for our virtual clinical trial. He has a great deal of experience in clinical trial development. I am PI on this study along with Carolyn Childs, who's a thoracic radiologist at Wake Forest. And we are conducting this project under the leadership of Edith Pisano, who's the Chief Research Officer at the ACR. We have the participation of 2 clinicians in critical care and emergency medicine, Rahul Kashyap and Suresh Agarwal. And as I mentioned, data scientists from the ACR, including Laura Coombs and Kendall Schmidt. So all together, coming together as a pretty strong team to kind of give the perspectives needed to make this happen. So what is a virtual clinical trial? Let's talk a little bit about that. Let me just back up here. So the approach to a virtual clinical trial is essentially to take an existing data set and do a hypothesis testing in a way that you would in a conventional prospective clinical trial, but doing this on this existing data set, then running the trial, accruing to the cohorts as you would in a randomized fashion in a clinical trial, and then doing an analysis on your outcomes. So for this virtual clinical trial, and in order to do this, you need to have a large enough data set so that you can ask the kind of varied types of questions that you would want to ask for a clinical trial, including information on outcomes, side effects, and medications, and so forth. So need the type of depth of data that we have in this registry to run this type of research project. So the way that we are approaching this is to first begin by selecting out the COVID-19 clinical diagnostic question to be answered, define the eligibility criteria, and then estimate the appropriate sample size, and then identify a population for study within the Medric data. Then once we've done that, we would randomize the de-identified patients to the diagnostic arms, and then analyze outcomes. The advantages to this type of approach is that you can ask questions that are not feasible to ask in a conventional clinical trial. So questions that we cannot address in a clinical trial for ethical reasons or logistical reasons. In addition, you're able to really draw from a large and diverse population, including all strata of socioeconomic, ethnic, racial, and geographic patients, as well as practice settings. And that's important because what you end up getting will be more generalizable to a wider number of practice settings and patient populations. And then you save in terms of efficiency. So it takes less time, far less costs, and less of a logistical expense than conducting a similar trial with similar numbers in a conventional manner. The disadvantages, of course, is that you're dealing with you're limited to what you've collected already. So you're limited to existing data. So you can only ask questions within the structure of what you've collected. In addition, while it's a benefit to have a large diverse population and diversity of practice settings, the downside of that is that there is heterogeneity in the way that patients are managed. So what you may have available to you for patient care in a rural setting may vary significantly from what happens in a urban setting. You might have in a community practice versus a academic or tertiary or quaternary care. So those type of things may impact what you see in terms of outcomes. That you may also see a different patient mix depending on where you are in the country, not only because of differences in demographics, but also complexity of care based on what the strengths are of a particular practice. So all of those factors really kind of play into the heterogeneity of the data that's in this set, and it's very important to understand that and control for that. In addition, there may be biases in terms of patient selection. So what patients that a particular center decided to upload may vary from site to site and kind of imparts a certain bias. And so we need to be cognizant of that and try to control and balance the populations of the cohorts when we go forward in a virtual clinical trial. The characteristics of our current registry is that we, as I mentioned, we have a longitudinal data. So starting from early 2020, it's a pretty large data set now. When we begin our clinical trial, we were expecting over 30,000 patients in this data set and rising. We have a number of sites that are participating in this trial, and data is coming in with more sites coming online over the course of this year. And as I mentioned, it's multimodality and it contains not only imaging, but also clinical data as well. So for our specific clinical trial, our overall aim is to identify radiographic patterns and or clinical factors predictive of poor outcomes in patients presenting with COVID-19 without identifiable significant risk factors. This was the aim that we came to after discussions with the clinical providers who are treating COVID-19 patients at multiple centers. We felt came together as a team to kind of think about what would be the most pertinent question to be asking at this time in the pandemic. And it's certainly been a dynamic process, how these patients have been managed and how this disease has been understood and treated. So what the question we're trying to ask now is basically, okay, we know that there are a number of risk factors that put us at patients at a disadvantage when they contract COVID-19. Can we identify those patients who have poor outcomes, who are coming to their presenting without known risk factors? And our hypothesis is that there are radiographic or clinical parameters that exist that can predict a poor outcome in an otherwise healthy adult presenting with COVID-19 infection. So our sub-aim 1 is to combine artificial intelligence classifications on chest radiographs, and we're partnering with a group at USD to do this. And these would be chest radiographs obtained during the acute phase of COVID-19 infection, and then look at the clinical data associated with those chest radiographs to look for predictors of clinical deterioration during hospitalization, ICU admission, supplemental oxygen or mechanical ventilation use, and complications such as pulmonary embolism. We know that these patients are at increased risk of pulmonary artery emboli, as well as sepsis and stroke or death. Our sub-aim 2 is to evaluate the impact of different treatment methods on patient course. So, in this area of treatment for COVID-19 is an evolving field. At the moment, we would be looking at steroid therapy, antiviral therapy, monoclonal antibody, and supplemental oxygen. Our methods, we are beginning by stratifying our patient characteristics to create cohorts with comparable demographic, socioeconomic, and clinical characteristics. And as I mentioned, we're going to kind of take into consideration the biases that we mentioned, as well as the heterogeneity in our data set that we're aware of, and really working to be sure that these cohorts are as balanced as possible. And we're really relying on our strong statistical team to help us with that. Then we're going to extract radiographic data using some existing tools. We have a couple of different collaborators. As I mentioned, we're one group from USC that we're planning on beginning with. And what they're going to do is basically segment out these areas of involvement in COVID-19 on chest radiograph, and then take those imaging characteristics and correlate, do hypothesis testing to look for features that correlate with poor outcome. And then after we've conducted a small pilot study on a subset of Medric data, we'll kind of go back to the drawing board, refine our hypothesis as needed, and then we'll run a virtual clinical trial. And that will be on a larger data set that's, again, balanced to be representative of, so that each arm of the virtual clinical trial is roughly balanced and equivalent in terms of its composition. And using that preliminary data from our pilot, we'll power a properly powered clinical trial that will be conducted based on our refined hypotheses from those early pilot analyses. So again, the steps: step 1, drafting the outcomes and predictors; 2, implementing random stratified split into a patient population into exploratory and confirmatory samples; conducting multiple exploratory analysis; and then locking down the preferred model, and then evaluating the replicability of those preferred models on the confirmatory sample, as I mentioned. So where we are right now, we have, we're in the process of actually locking our cohort of patients. We are also will be in the near future beginning to begin our radiographic data extraction and feature analysis. At the moment, we are finalizing our methods and our protocol, and we'll anticipate having results most likely by our first results by the late spring, early summer. Once we have conducted the exploratory study, we'll be then conducting the confirmatory virtual clinical trial. So we're kind of at an exciting stage of this very novel approach to conducting clinical trials. I'm very excited to see how this goes. So I just want to thank you for your attention, and I'd like to welcome any questions you may have at this point.
Thank you, Sharon. That's very exciting. And I think this shows a great use of the Medric data and community. We do have one question posted that I'll go through, and then if others want to add in, we'll look forward to a discussion. So there's the one posted that says, how does your AI algorithm handle multimodal images, CT, ultrasonic, X-rays, etc.? Is the implementation open source so I can look at the implementation, the algorithm? These are the algorithms used in running your clinical virtual trials, I would assume.
I think that's an excellent question. So the AI algorithm that we're going to be using to start coming from USD is going to be just on chest radiographs. Yes, it will be open source. I think it's really important for validation in science to have these tools open source, and of course the data is open sourced. So that will be available once we're completed. And I think it's a very important question to next look at CT, and we don't have a tool for that at the moment. Something that we're looking into now, but I think that'd be a really interesting application, not only for predictors of response to therapy, but looking for tools to differentiate COVID-19 infection from other disease processes that can look kind of similar. So, as we kind of go forward living with COVID in our community, now that becoming part of our differential diagnostic considerations in cancer patients and patients with other disease entities, it may be helpful to have tools to help us differentiate. So I think a lot of interesting questions could be asked on this data set.
Thank you. Let me see, is that, okay. Others, please post your question. So I think you said it's open. So just so the audience knows that Medric is working on making parts of its GitHub. Some are for the investigators developing them now. Others will be then posted in the open format. So please keep checking the medric.org website as that information becomes available. Medric has a variety of algorithms, both on detection of COVID, distinguishing COVID positive versus negative, as well as looking at severity of the COVID patient, similar to what Dr. Katz pointed out about looking at them in the hospital for different conditions over time. And hopefully also then for surveillance. So we have about 24 algorithms of Medric out there. So please keep an eye open for those. I have a question, Dr. Katz. So I think this virtual trial methodology is of great interest to many users of Medric, and many users or future users of Medric data will be developing their AI algorithm. When you have a virtual trial, and let's say you have your first COVID positive or negative tool, you run through it, will that be available for others to run their algorithm through it to see if similar, for a given hypothesis, for different AI tools, meaning they are after the same task, but they come from maybe 3 AI algorithms from 3 different research groups, would they give the same clinical result relative to your initial hypothesis?
So if I understand your question, are you saying will we be providing our cohorts, or so that other
right,
tools
so
so
other AI developers can run on those cohorts.
That's a good idea. Yeah, we hadn't discussed that, but I think that's an excellent idea for validation. And so that we can compare equitably. Yeah, no, I think that's an excellent idea. We hadn't talked about that, but I'll definitely bring that to the group.
Okay, great. Because I could see running, if we have 3 different AI algorithms all going for the same task, that then, in a virtual clinical trial, one is, say, try to identify a cohort, what is the specific task of that AI algorithm? But the other one, if it's characterizing the chest radiograph for your clinical endpoint in your virtual clinical trial, it'd be interesting if you get the same clinical endpoint too.
Yeah,
so I look forward to that.
That's a great idea. And I think that's one of the great things about this, about Medric is that it really kind of enhances collaboration in a way that I think it's unprecedented. I'm excited about it.
And do you have questions? I think this is a great opportunity for the ACR, the CRP7 virtual clinical trials. Do you need volunteers? Do you need other clinical questions? Is your team expanding?
Yeah, excellent question. So we're starting with this first question, but I actually really would. That's another excellent point. I actually would welcome thoughts from the community on what other important questions we need to address in our database once we've taken the time to kind of statistically control these two cohorts and balance them and create these sets. It would be great to maximize that. So yes, I actually would welcome anyone who has any thoughts on ways they'd like to use this data to reach out to us. We'd be very interested. I think we're going to just start with this first project, and I think it's our first time doing this. But once we've done that, I think yeah, we really welcome next thoughts on where to go from here.
Are there other questions from the audience? Here's one. It's Cynthia, must be a company, makes available standardized synthetic data cohorts for cross-testing of algorithms on the exact same data set. That's interesting. If you can post that website, people can go there and look at it. I think it would be interesting to see. This seems related to clinical trials, the Cynthia, SYNTHEA, for folks who don't have access to the chat. It'd be interesting how the standardized synthetic data cohorts are similar to actual clinical data. And one would assume those cohorts need the breadth of the distribution, similar to Dr. Katz was going over between her pros and cons of virtual clinical trials, that the data set has to have enough cases as well as span the type of diseases as well as the normal. So,
right.
Oh, it's from MIDRC. Great. Okay.
Actually,
this is what we want.
And I didn't bring that point up, is that we're collecting, Medric is collecting not only positive cases, but also negative cases as well. So patients who are COVID negative as well as positive. And for our initial trial, we're looking at only positive, but for those who are asking questions that are going to involve, pose questions that would require controls that were negative for COVID, that data will be there as well.
Okay. Other questions? I think we've covered it. I look forward to perhaps having you back in a year, where we now have clinical decisions or clinical findings based on the virtual clinical trial methodology.
Thanks for having us. Yeah, and please, again, anyone interested in reaching out, giving you your thoughts on directions, more ideas, and where we can go with this would be much appreciated. Thanks for having me. It was a pleasure.